Good morning, and thank you for coming to 2024 United Leukodystrophy Foundation Family Conference. And I would just like to give you a quick disclaimer that the sessions are being recorded and it will be posted on the United Leukodystrophy Foundation website and YouTube channel. It is a great pleasure for me to introduce our speaker today, Dr. Bonkowski, presenting Zebrafish, Mice, and More.
Good morning, everybody. Hopefully everybody's awake and not too cold. I'm going to keep my jacket on for right now. So I'm going to talk about two things this morning. The first, I'm going to give kind of an introduction to clinical trials because I think understanding how we get to treatments really is helpful because then all the research that gets done in animals and different models is what kind of gets a clinical trial to take place. And then I'll also say that the information I'm presenting, I've tried to boil it down because it's a complicated field, and trying to understand like how a clinical trial works and how all these different research models works is super in the weeds, but hopefully I've kind of captured some of the key elements here. And there's plenty of time for questions as well, so please raise your hand or just speak up if you have a question. So I think one of the challenging things as researchers, as physicians, as patients, as families, is that the next great thing is always around the corner, and I think it's important to keep that sense of hope, but also to be realistic that there's a lot of challenges. So I think this quote from Martin Luther King Jr. is great because we always face disappointment day to day, but we have to keep our hope alive that we will get to a better place. So I have various disclosures, nothing really related specifically to what I'm talking about today, but just so everybody's aware of these different things that I'm involved in. So getting to a treatment, so getting to something that you actually use, get a medicine prescribed for a disease, takes many years and millions to billions of dollars. The cost from when you have an idea for finding a drug to actually getting it to a person can take several decades, and that's if things go well. And so this is just for like one drug for one disease, and so of course we're talking about many different diseases. And so the challenge is really high. It's very expensive. At any point, something can go wrong in any one of these steps, and the entire process can get frozen or halted. So to get there, I'm going to talk first about clinical studies, about clinical trials, because that will then help us understand why we use these different research models. So a clinical study is something to understand if a treatment or an intervention makes a difference. And the reason to go through this kind of complicated series of steps that I'm going to talk about is because you really need kind of a scientific, logical approach to doing this. There's no guarantee that something's going to work, and you have to really go through these steps to not fool yourself or not fool your patients. There have been examples, even in the not too distant past, of where we, the scientific medical community, have kind of gone down a path where the data wasn't great, but our hope was so high that we kind of fooled ourselves. So if you don't go through this process in a really rigorous way, really you're just selling snake oil. Like, it's no better than just kind of guessing and doing things that don't make a difference. So there's different kinds of clinical studies. The one I'll talk about most are what are called clinical trials, which are kind of the rigorous series of steps to go from having a treatment that you hope might work and ultimately getting it to patients. Or the other kind are what are called observational studies, where you kind of look at what's happening in the real world and see if you spot differences that are kind of hidden in the noise. So for example, if there's a bunch of people in California eating avocados and they're all super healthy, you're like, oh, maybe avocados are really healthy. So that would be kind of an example of an avocado study or an observational study. So an observational study doesn't have, you're not doing something actively, you're just kind of watching what's happening and seeing if it kind of informs something that you didn't realize was going on. The most common kind of observational study that you'll often hear spoken of in some of the discussions and that many of you may have participated in is something called a natural history study. And these are really important because there may be clues in what's already going on to understand a disease or understand a disease process. It can kind of show why maybe some people are doing better or some people are doing worse, and then when changes occur. And this information, it's very boring in some respects because it's something any patient or any family person could tell you, but when you put all the information together, it paints a picture, and that picture is what we then take to the FDA or take to the government and say, this is what it looks like, and this is why the drug can make a difference. And without that information, it's very difficult to get FDA approval for drugs. And so there's been examples, even in the past couple weeks, of where without natural history data, the government agencies are unwilling to support a new drug because they just aren't certain that it's actually making a difference. Also, if you want to know if your drug makes a difference and you don't know when the disease gets severe, then how can you know if your drug is really working? So it's a critical kind of ground piece of data for any kind of clinical trial. So clinical trials, there's lots of language and lingo around it. There's different phases or steps, and it's a little bit complicated. It's taken me many years to kind of get the hang of all this, and so hopefully it kind of makes some sense as I'm talking about it. Each of these phases is kind of a stepwise progression. You need to go through the steps one by one, but there's like this kind of a classical way to do this, but of course there's different setups, of course, at work. A clinical trial is expensive, like in the millions of dollars to do. For a drug company, they're supported by investors, and if the investors get cold feet and suddenly realize or are worried that they're not going to make a profit, they'll pull out of the clinical trial because they're losing millions of dollars. A clinical trial is very slow. The paperwork is just immense. I see some people from our clinical trial team kind of hiding in the back there, and they know what that paperwork looks like. It is like it's like months of time and many different moving pieces to put it all together. Because the clinical trial is expensive and difficult, if you go in with bad science, you're wasting everybody's time, you're wasting everybody's money. And so that's why the research that we'll talk about in the second half of the talk, you want the research to be really good. And also the observational studies, if you kind of are just flying blind without knowing what happens during the disease, it's also not worthwhile doing a clinical trial. So a clinical trial is a big investment on, of course, the part of the physicians and researchers and their clinical research teams, but really a lot of the burden is carried by the participants and family members, and that is non-trivial. It is a big investment of time. It is often a hassle, like in terms of travel, in terms of blood draws and MRIs and other things you might not be doing. And so really, you're doing it out of hope, in partially for yourself, but you're also really kind of paying it forward to the community. So it's a chance to help others. It can help fight these diseases which are a scourge. And ultimately, we hope that they succeed, but there's no guarantee that a clinical trial will be a success. There are reasons not to join a clinical trial, and so that's important to think about if you're asked to participate. If the trial has more requirements and more steps that you really can't handle, if it's going to destroy your marriage, if it's going to be impossible to do the travel, don't do it. It doesn't. There are reasons not to do a clinical trial. There's even though clinical trials are reimbursed typically in terms of travel, in terms of lodging, time away from your family or from your other kids, that's a real thing. And then there are stupid clinical trials out there. So if it is a stupid clinical trial, don't do it. Usually your physician or the researcher you're working with can kind of give you advice. But if you're not certain, check around, check with other families, check with investigators at other institutions to make sure this is a good idea. So who is doing the clinical trial? So someone's paying the money for the clinical trial to happen. Sometimes it's the pharma company paying the money. Sometimes the government, like NIH, like we just heard, will support a clinical trial. There's other things like some philanthropy organizations will put together clinical trials. So it's important to understand who is paying for the clinical trial. Then you have the research team that's doing it. The research team ranges from what's called the PI, the principal investigator, and then the people that you'll be seeing day to day are usually the clinical research coordinators, physical therapists, occupational therapists, neuropsychologists, are all the people kind of doing it. There should be physicians involved in a clinical trial. The whole study team, like as I just mentioned, the whole trial is overseen by a variety of regulatory agencies, and that's why the paperwork is immense. The paperwork is immense for a good reason, because you don't want crazy things to happen, but of course it makes it more complicated. And then each institution has something called an IRB, or Institutional Review Board. The IRB's job is to make sure that the clinical trial idea and the way it's being proposed is safe to the people participating, and it is going to provide a benefit to participating in. And so their job is to really make sure that it's going to be a good idea for you as a participant. They can't oversee every single part of the trial, but they have a very key role to make sure it's safe. And so if you're ever involved in a clinical trial and you're having difficulties, the IRB is there to help you out as a participant. So the IRB, each institution has an IRB. There's also IRBs that work across multiple institutions. That's becoming more common now. It's required for any research involving people. When you're asked to participate in a clinical trial, you do sign various consent and assent forms. They tend to be like fairly thick stacks of paper when you're signing it. On the one hand, the information in the consent forms, it's like a lot of information. A lot of it's kind of like not super relevant. On the other hand, when you sign these forms, you should feel like moderately comfortable that you understand what you think you're getting into. It is important to do this, but it does take a lot of paperwork. So with any clinical trial, there's lots of kind of big questions to ask. And so one of the questions, of course, is that, are you being asked in the clinical trial to do stuff that's like crazy or unsafe? And so the IRB is supposed to prevent that from happening. One issue that's come up in the past is that the protections we have here in the United States or in Western Europe are a lot better than, for example, in some places in Africa. And so some drug companies have tried to get around some of these restrictions and costs by doing clinical trials in Africa, and that's really been kind of shut down a lot. One thing that we've run into even, for example, is that if we have families who have less means, like for travel, or speak a different language than English, that it's actually quite difficult for us to help them participate in a clinical trial. And that's really a challenge that we're all facing, to make sure that we have equal access to be in a clinical trial. And then one thing that has sometimes happened is at the end of a clinical trial, particularly if it's gone well, what happens then to make sure that the people still get the medicine? And that's not always like defined at the start. Everyone's very aware of that problem, but it's a little bit tricky sometimes to make sure that the drug keeps coming to the participants at the end of a trial. So this is a overview schematic of the different phases of a clinical trial. And so here at the far left-hand side, preclinical, this is all the research steps that I'll be talking about in a few minutes. Really, without having good science, going into a clinical trial doesn't make any sense. And these are kind of the classical phases of a clinical trial. So at the end, you're trying to get to what's called phase 4, where the drug can then be prescribed. And so this is kind of phase 4 is everything that happens after the drug's been approved. And so medicines we use like amoxicillin or that's like the only example I can think of. So something your pediatrician can prescribe for you is something that has gone through these earlier phases, is now in phase 4 and can be prescribed by any physician. In phase 4, sometimes there are still requirements that the government or FDA will require. So for example, they still want to make sure that the drug is safe, and so they'll continue to kind of monitor what's happening with the drug after it's approved. So for example, with the COVID vaccine, for example, because it was an emergency, it went through this process very quickly, but now it's still being kind of monitored for any kind of safety effects now that it's been approved to be marketed and prescribed. But then to backtrack, so phase 1 is kind of the safety step where you're making sure that the drug is safe for people, and so oftentimes that step is where the drug is given to healthy volunteers at very low doses, make sure there's no surprises. In phase 2, there's more safety steps, often looking for side effects and sometimes seeing if it works. They'll start to give it to people with the disease. And phase 3 is really kind of the key phase where the drug is compared against either a placebo. A placebo means like no drug, or against another, or the drug is compared to the kind of the standard treatment to see if it works. And so at any of these steps, the entire trial can be stopped. There's the drug company can stop the study, the government can stop the study, the IRB can stop the study if there's a problem at any of these steps. So for example, if you start giving it to healthy volunteers and they start having heart attacks, you stop the study. If you're giving it in phase 2 and you see that people have effects on their liver, you stop it at that phase. If you start in phase 3 and you're going along and there's no signs that it's working at all, you stop the study. So it can stop at any of these phases. And so sometimes you'll read in the news about drugs at different phases where it gets stopped by the company or gets stopped by somebody and doesn't move on to get marketed. And so it's a huge disappointment. And you may have spent millions of dollars to get to one of those phases. And I'm sorry, there's a question there. So I'll repeat the question so everybody can hear it. So the question is, when does the patient become involved? And so the patient can become involved at different steps. So sometimes patients become involved in the preclinical stages, even where like a blood sample or a sample of tissue is needed to see if the drug is changing kind of the biochemical markers, but that's not really what you mean, I think. So usually phase 2 or phase 3 is where participants with the disease will be signed up for the study. And phase 1 is usually done in healthy volunteers. And so it's usually phase 2 or phase 3. Now sometimes companies are doing more, not tricky things, but more complicated things. So for example, like a phase 1-2 study where they're kind of combining different steps, or they'll do like a phase 2-3 study. So it can be a little bit more complicated than this outline, but this gives like a general sense of like kind of how it's laid out. So usually if you're being asked to participate, either it's a phase 2 or a phase 3, and in the consent process, you should be told what's being tested, like are you just being tested for safety, or are you being tested if it works? And then you should also be told what's your chance of receiving the drug. So for example, many phase 3 studies will compare the drug to a placebo, and so you want to know like what is your chance that you're receiving the drug versus the placebo. From a participant, from patient perspective, everybody wants to get the drug. But from a scientific perspective, the scientist wants to be able to compare the drug versus not drug, and so we want people not receiving drug and people receiving drug to compare to each other. So there's kind of this tension for how to achieve that. So there's various complicated types of clinical trials that we've designed now to try and give everybody a chance to get the drug or to build in some way to do that. It's a whole thing. Oh yeah.
being developed specifically for rare disease? Is it more common? I work in cancer, and in cancer, the phase 1, you don't go into healthy volunteers, you go right into patients. And so I thought that in rare, that often was the case as well, but I don't work in rare, so.
Yeah, so I'll repeat the question. So the question is, in some diseases like cancer, oftentimes there's not a phase 1 because the drug might be dangerous, for example, or have like significant side effects that they know about, and it'll go straight into a phase 2 or 3. And so that's absolutely correct. Again, this is the classic example, but depending on the drug and depending on what's known about potential side effects, and depending on the disease, they may skip straight to the phase 2 steps to look at a drug. There's also different pathways based on the rarity of the disease. How rare it is means that it may be very difficult to do a classic phase 3 study, and so that's another example where it's how this gets set up is different. Also, depending on the severity of the disease, or the like an example of where this didn't really happen was like with COVID, where it was a public health emergency and they just had to basically go as fast as they could into kind of like a phase 4 delivery of the medicine. So there's huge variability in how this process works. So just to remind ourselves, so preclinical, this is all the stuff that happens before it reaches patients. So does the science make sense? Phase 1, is the drug safe? Phase 2 usually is kind of classically figuring out some sort of dose and some sort of way to measure the effect of the drug. And phase 3 is efficacy, does it work? So kind of the simple, this is like a simple, if you're taking a test after this, this is what you'd need to memorize. So then there has to be, so different countries and have different regulatory agencies that oversee medicines and drugs. So in the United States, that's the FDA, the Food and Drug Administration. And in Europe, it's the EMA, the European Medicines Agency. Those are probably the two agencies currently in the world that have the kind of the biggest effect on marketing. There's also obviously administrations like in Japan or in other countries around the world, but these are the two that researchers and pharma companies are really paying the most attention to as they try and bring their drugs to market. And they have slightly different rules and different roles, and so there's a lot of thought about how to work with these agencies, because if the FDA or the EMA says no, then that shuts the whole process down. So the FDA approval process is super complex and confusing. I don't really understand it. So I mean, there's lots of paperwork and forms. There's also there's steps that you're supposed to go to, but then there can still be pressure from outside groups, whether it's a senator or a family group, and that plays a role in the process. So really how it should work, it should be proving that the drug is safe and doesn't hurt people. And then the second thing is really it should be effective. Like if the medicine's not effective, then why give it? Now it gets a little bit complicated, because what if it's effective in 20% of people with the disease but not in 80%? And so that gets into statistics and math and kind of judgment calls. There's also a lot of debate about, like, what if a drug helps you live 6 months longer but costs $5 million? And so that's also like a complicated thing that the FDA is kind of facing, and it's been in the news sometimes. And then getting back to the question here, there's different types of approvals for different categories of medicines. So for example, there's emergency use approval, and that's what happened with COVID, because there's the risk of like millions of people dying, so they went through the process much quicker, kind of looking mostly at safety. Excuse me. There's also a process called an IND, Investigation of New Drug, and that's a whole kind of subset of processes where there's a new drug showing promise. Maybe it's for a rare, very severe disease. You're getting special permission to give that medicine. And then orphan drugs, and this is often what we fall to in the leukodystrophy space with rare diseases, and there's a whole kind of separate sub-process for how drugs for orphan diseases can go through the approval, because it's recognized that the barriers are different for diseases where you only have 10 or 20 people affected. So this is kind of a snapshot of the FDA process. There's kind of a review meeting, and there can even be kind of like pre-review meetings where the people doing the study come to the FDA and say, here's what we're thinking we're doing. What do you guys think before we come to you officially? And the FDA can give feedback like, oh, that's a good idea. You've got all your pieces lined up, or oh, we're not going to accept that data. You need to do it differently. Then a new drug application is put in. The FDA reviews the application, and they have a specific timeline to finish that review. And then at that point, if it is approved, there's a drug labeling process where the FDA says, you can say the drug is used for this under these conditions, and that drug labeling then impacts how insurance will or will not cover it for families. And I'm sure many people kind of have dealt with this here, where you're trying to get approval for a drug, but it's off, quote unquote, off-label, and that can be very difficult. The FDA continues then afterwards to make sure that the production of the drug is done safely, and so they'll inspect the facilities where the drug is made. And then there's a official drug approval letter that's given out by the FDA. And then finally, kind of coming off the right-hand side of the slide, there's kind of post-marketing tracking of different drugs. And so certain drugs have more requirements for post-marketing follow-up. So for example, a lot of the gene therapies, the FDA is requiring like 10 years of follow-up after approval to make sure that there's nothing like higher rates of cancer or something in people who receive those medicines. So in a clinical trial, if we're lucky, we might test several hundred people to see if it works or not. But then in the real world, we may be giving this drug to thousands of people. And so at that point, very rare events might start to happen that we hadn't realized with the clinical trial. So for example, if you have a drug and you do a clinical trial with 500 people, those 500 people do fine, that's great. But then in the real world, you give the drug to 1,000 people in that first year, and the risk of stroke is 1 in 1,000, suddenly you'll start to see people with stroke from that drug. And so that's an example of how you have to continue to track the drug after it's approved and on the market. So just to wrap up this part on clinical trials, so observational and clinical trials are both very important for how we do this. The observational thing that we often talk about are natural history studies, and so if you're asked to participate in a natural history study, it really is a key part of this whole process. If you're asked to participate in a trial, make sure it's with someone you know and can work with well, because you want to participate in a trial that makes sense, but you shouldn't be participating in something that's hurtful to you or to your family or to your child. Make sure you understand the details. Think about how you're contributing and making a difference, like you really are part of the whole process. And then realize that it's extremely difficult to see the future. Things are much clearer in retrospect, and so you always want to be hopeful, but also be realistic about what's going to happen. So we're going to transition here to talk about research models, and I'll just stop for a second and see if there's questions about clinical trials.
Thank you. I was just curious if the whole drug approval process, which I'm sure, like you said, is very complicated, does that happen outside the clinical trial process, or do you start that in one of those phases of the clinical trial when you start to see, or if you start to see any, significant improvements there?
Do you mind just saying that again? I'm not sure I understood your question exactly.
The drug approval process through the FDA, does that start after the completion of or is it when you start to see some improvements, can you start that? Can they go simultaneously, or is it like you got to do this and then that?
Yeah, so what will happen so a clinical trial is being set up, and usually even in the very early stages of a clinical trial, like a phase 1, 2, if the company's feeling, if the company or the researchers are feeling like optimistic, they'll start to approach the FDA in kind of these pre-meetings to say, here's what we're doing, here's the outcomes we're looking for. Is this something that you, the FDA, will accept or not? Because the FDA could, someone could go through a clinical trial and could measure walking, how well you can walk as a outcome. But the FDA could say, well, we don't like that outcome. Instead, we want to know how well you can talk. And so the drug company doesn't want to be surprised by the FDA, and so they'll often make sure that the outcomes and the way they're doing the study will be acceptable to the FDA. And so oftentimes they're slightly off kilter but occurring in parallel. Yeah. And then other questions about the clinical trial process? Nope. Sure. So the question is about the need for the drug from kind of how common the disease is. And so there are specific pathways for orphan diseases that are slightly different than the classic steps, because the FDA has come to recognize that the rarity of disease means it's difficult to, for example, if you only have 2 patients with the disease, you're not going to have statistical ability to test and compare the drug, and you're also going to, so that's like kind of one example. Another example is if the disease is so severe that unless you give the drug right away, they're going to die in 6 months. There's also the kind of that recognition. However, the FDA, this process of kind of moving how the FDA thinks about drug approval processes, they're still learning how different it is in the rare and orphan disease space compared to kind of the very common disease space. So I think there's still some tension there about those kind of questions. Yeah. So the parent and family group involvement in drug approval and clinical trials is very important, and so typically, as a clinical trial is happening, either the researchers or the drug company will start to work with the disease community in preparation for approaching the FDA, because when the FDA goes through the review process, there is kind of an open comments session where families and patients can speak directly to the FDA about their experience, and those do weigh into the FDA decisions. And that's where another example of where it becomes very challenging to decide about the right thing to do. So for some of the Duchenne muscular dystrophy is an example of a disease that's very severe. It's a orphan disease in terms of total numbers of people affected, and the treatments are not that. The data on the clinical trials wasn't that impressive, but because of the need, the drugs got approved, but they're quite expensive. And so like what's the right thing to do? Like, it's not totally clear. And so hopefully drug companies are approaching like the ULF or individual family organizations to be involved in the process in approaching the FDA. Yeah. And so I think on the one hand, we want to help the FDA realize the right thing to do. On the other hand, I think we need to be aware that sometimes there are drugs that are bad ideas. And so sometimes if it's not working and you're going to be asked to pay a lot of money for it, maybe that's not the right thing to do. And so it's challenging for sure. So I'll talk now about, so we'll kind of shift gears here now and talk about research models in leukodystrophies. So why are we doing research? Like, what's the point of all this? Like, why are we spending so much money doing research? So there are really 4 big reasons. The first is we need to understand the biology of the disease and we even need to understand like the basic biology. So if you don't understand like what a lysosome is doing in a cell, it's going to be really hard to understand metachromatic leukodystrophy. So you need to know like what is the lysosome, what does it do? The second thing is we need to understand what the disease is doing, like what is it messing up? Because if you don't understand what the disease is messing up, like how can you possibly fix that? So like if you have a house and there's a bunch of water in the basement, like you need to understand like where's the leak coming from, and so like where in the cell are things not working. Another thing we do in research then is like you have an idea for a treatment, and you need to see like is the treatment or idea that you have making a difference in the biology, and is it making a difference in the disease model? So you're kind of understanding like what is the treatment doing, or like what is your idea for how this is going to change the biology. And then of course, in the research, we're often trying to discover treatments. So these are kind of the 4 big reasons we do research to try and get us to the point of getting a treatment or a drug to a patient. So why do we do this like in these, I'm going to be talking about some different animals. You can be like, why do this in an animal? So first of all, working with people is very expensive. Like it costs lots and lots of money. And so anything you can do in a less expensive animal model is great. The second thing is there are experiments that are very difficult to do in people, but that are pretty easy in a mouse or in a zebrafish. So for example, zebrafish are transparent, so you can just like look at their brain under a microscope, whereas you can't do that with a person. And then finally, there are lots of things we do to our animal models that are not ethical in people, right? So like, so that we don't want to do unethical experiments on people, and so that is a reason to use these animal models. Because of all the complexities of a clinical trial, you do not want to start a stupid clinical trial. And so so you want the science to be really good before you start a clinical trial. And so the research really kind of lays like this super important groundwork for what's going to happen next. So this is a famous painting from René Magritte, I think, and it says, "This is not a pipe." But of course, so it's a picture of a pipe, so it's not technically a pipe, but we look at it and we're like, oh yeah, here's a pipe. So this is what a research model is. It is not the same as, so if we study MLD in a zebrafish, it is not MLD in a human person, but it is a model for what we're seeing. So just the same way this is not itself a pipe, but it's a representation of a pipe, and we kind of understand like how to think about it. And so that's like one important point, is that that a model is a representation of disease, but it has kind of, obviously this has a limitation. You can't smoke this picture, but it helps us kind of think about a pipe. The other big point I want to make is that every single model has limitations. Whether you're working with zebrafish or mice or even people as your model, they all have pros and cons, and those are significant. So this is like an example that I thought was really a great example. And so this was a study that came out maybe now about 8 or 10 years ago, where they looked at the inflammatory response in mice and compared it to the inflammatory response in people. And the one-to-one match between mice and people for this inflammation response was 50-50. So it's basically you could have flipped a coin and just done just as well for guessing the inflammatory response. And so for a long time, mice have been kind of the standard in the field, and mice are great, but we should also recognize that whatever model you have to think about, like, is this model a good model for the disease you're studying? So, and I guess this is my third major point, is that the genes that we have and the way our biology works is really the same across most species. So whether you're working in humans and mice and chickens and zebrafish, there's like about 10,000 to 11,000 genes that are all basically the same, and the biology is basically the same. And so if you're working on that group of genes and diseases in biology, like you just choose the model system that's going to help you the most. Now, of course, people are not mice. Zebrafish, live in the water. We don't live in the water, so there are like real differences, between us. So like studying gills in zebrafish is not going to translate into studying like lung function directly in people, but understanding how, for example, the white matter in zebrafish, which is conserved with white matter in humans, that makes a lot of sense. You really need to think like, what are we studying? Does it make sense? So there are many different research models. I've tried to boil it down to the ones that really are kind of being used and tossed around most commonly. So we already talked a little bit about natural history studies. So this is where we kind of look at what's happening in the real world and kind of see what's happening to groups of people. And that often is very informative. So cell models are often with something called fibroblasts, which are skin cells, where you look at just at kind of individual cell types and kind of study what they're doing and how they respond. Something called organoids, which are cells that have been grown and to grow into kind of like little mini organs, where they're like little mini brains, for example. Drosophila and C. elegans I've kind of grouped together here. Drosophila are fruit flies. C. elegans are roundworms. These are kind of the most simple animal models that are commonly used. Zebrafish are a more complex vertebrate animal model than mouse models. Then finally, even humans kind of serve as models. And I'll talk about all these in a little bit more detail. So this is a very rough color scheme for the different models. It's obviously, this is like me on Tuesday night kind of working on this, so it's a little bit made up, right? So it's not like, and so green is good, red is bad. So let's start at the bottom here. So humans, obviously something we discover in humans is going to be very relevant to other humans, but working humans is pretty slow. Like, you have to convince them to come in to do the work, so it's going to take a long time to do this. The cost of working with people is very high, and so it's very expensive. Of course, anything you discover in humans, like that's going to be very powerful because you can just take it right over and start using it. And then the same thing here, like, does it translate? Like, yes, of course, if it works in one person with a disease, it's very likely to work in another person with a disease. So mice, so they're a mammal, they're a vertebrate. They have a lot of the same genes, so they should translate pretty well to humans, but in fact, mice are pretty different, so there's a lot of differences between mice and humans, so it gets kind of a yellow. Mice, kind of a medium speed, so they get orange. Mice, unfortunately, are also pretty expensive. The cost of taking care of mice, of hiring a vet for the animal facilities is expensive. However, like mice, you can do lots of great experiments, so you have a lot of power of discovery, and then we'd like to think that because we've studied mice for so long and have so much data, you can often take a discovery in the mouse and move into a human study. Zebrafish, again, it's a vertebrate. They live in the water, so a lot of differences, so relevant to humans. They're pretty fast. They grow fast. You can do lots of experiments very quickly, so very powerful with their speed. Their cost is kind of intermediate. It's much less than mice, but it costs more than like Drosophila. Because you can do so many experiments, they're very powerful for doing research. But then the stuff you find, like, is it going to translate directly? You probably still need to take those discoveries and kind of confirm it in something like a mouse or some sort of human-related cell line. Drosophila and C. elegans, these are some of the more basic animal models. And so there's kind of concerns like how relevant will be for humans, and some of the biology is very different in Drosophila. But the speed is super fast. Like, they're an amazing animal system to work with. You can just do like these amazing experiments super fast. As you might imagine, they're pretty inexpensive to work with. And your ethical concerns are lower working with fruit flies. But then they're super cool. You can really discover these great things working with Drosophila. But then, like, can you translate it to a human? Well, like, you're probably gonna have to take that discovery and go confirm it like in a mouse or something. So organoids are human-derived cells that then have been turned into little mini organs, and so you can like grow like little mini brains in tissue culture. They're cool, so they have human relevance, but they haven't like completely transformed everything like we thought they would 10 years ago. They're pretty fast to work with, but they are moderately expensive. Things that you discover, because they're human-related, should be able to move over, like you should really be able to make good discoveries, but then they just haven't so far, like I just feel like they haven't really like transformed the field the way that we thought they would. So cells are like individual types, what are called cell culture, where you just grew a bunch of cells in plates, like little petri dishes. Usually people are using something called fibroblasts. So of course, something that you discover in a fibroblast in skin, is that going to be the same as how your brain is working? I mean, maybe, maybe not. So sometimes it's hard to know how relevant it will be. On the other hand, like super fast to work with, low costs. You can do lots of discovery, but again, it's just like individual cell types, and so, you lose the complexity of it, like working with a whole animal. And same thing with translation, like is a drug that works in a bunch of cells really going to work for a whole animal? Sometimes, sometimes not. Then observation studies are where we go do natural history study, obviously very relevant to humans. The work is kind of medium fast, the cost is pretty low, but you are really limited with your discovery with what's already going on, and sometimes it's hard to translate what you see into like real world actionability. So just natural history studies have a certain place, but they also have limitations. So this is an example of like natural history study. You're trying to use that information that's already out there based on what's already happening. You really rely upon big databases or tracking individual patients. So it's really, if there's a lot of information out there, you're in great shape, but if there's not information, you have trouble. So for example, a great kind of example of this is until recently we didn't have very many ICD codes for leukodystrophies, which meant it's like extremely difficult to know what was going on with like vanishing white matter disease or Alexander disease or Pelizaeus-Merzbacher. So it just meant that you couldn't like see what's going on across the country for these people. But you can also see like different patterns if you have good data sets. So for example, this is a data set that we looked at a few years ago where we looked at all the different leukodystrophy patients across the country, and we saw that people were diagnosed in certain kind of clumps, and the clumps where people were diagnosed were based on how close they were to a major academic center. So you're like, so unfortunately, like if you lived like in the Midwest or the Deep South, like your chances of being diagnosed were lower because you didn't have good access to a medical center. So it really kind of highlighted to us that it's important like to give better access for people for diagnosis.